Bismillahirrahmanirrahim. My name is Aisha Bibi. My discipline is BS Biotechnology and my roll number is 3. Today the topic of my presentation is lab test for the cleanser. There are some tests for the products of the cosmetics. The first is preservative effectiveness test, stability test, microbiological stability test, packaging stability test, safety and toxicological test, compatibility testing with packaging, and then pH testing. First of all, preservative effectiveness test. Cleanser is aqueous-based product and it contains 90% water, and this provides environment conducive to microbial growth, and also some vital growth ingredients for the microbes are present in the formulation. Ingredients like surfactants, dispersants, emulsion, rheology modifiers, and enzymes, and they can also decrease the efficiency yield of the product and can be dangerous for the human health. Antimicrobial preservatives are added to products to prevent or limit microbial contamination, and these preservatives are for the normal condition of storage and use. The efficiency of the antimicrobial preservatives may be enhanced or lessened by the active constituent of the preparation, or by the formulation in which it is incorporated, or by the container or clear being used as a final packaging material. The test method must be qualified for the product under evaluation to ensure the correct diluent is used in assays for surviving microorganisms. For this, we take 2 plates product in which in the first we inoculate the microorganisms and held 28 days at room temperature, and we examine the microorganisms plate with the second duplicate plate which held with the product. Then come to the stability test. Stability test provides evidence on how the quality of drug substance or product varies over different environmental factors like temperature, humidity, and light at the given time interval. Microbiological stability test. Contaminants usually come from 2 different origins. The first is during production and filling. The second is when the consumer or customer use the cosmetic products, and for that moment the different microbial things can come from the hand or the environment to the cosmetics. Then come to the screening test. There are various easy testing kits available on the market. For example, dip slide or plate counts. Dip slide test is used to check the presence of microorganisms in liquid, and dip slides are incubated at 30-degree temperature for 48 hours when they are dipped into the sample to ensure the result. Come to the plate counts. Plate counts are the test in which we inoculate the microorganism at one plate, and we can compare one plate with the second. And these provides a quick and semi-quantitative result whether our cosmetic product is significantly contaminated or not. Sampling and evaluation of the result is simple and can be performed also by personnel without any microbiological training. Then quantitative test. Quantitative test determine the actual count level of bacteria, mold, and yeast in a cosmetic products. These tests are very sophisticated and laborious and can be performed only by the professional microbiological testing laboratories. Typically, methods for isolation of microorganisms from cosmetics products including direct colony count and enrichment culturing. Then packaging stability test. Packaging can directly affect finished product stability because of interaction which can occur between the product, the package, and the external environment. For example, product constituent may be absorbed into the container or may chemically react with the container. In addition, the container may not full protect the product from the different environmental condition like oxygen and water vapors or volatile product constituent may evaporate through the container. Glass test. Glass is the most inert material and doesn't react with the cosmetic products in any way. For this reason, all the tests which we perform are performed in the glass and actual packaging. In this way, you can determine if the cause of product failure is a formula or the package. Weight loss test. To determine evaporation means the water loss can be from the product, and water loss evaluation is one of the most important tests that must be conducted. This testing performed in the actual packaging with the gap torque to 100% of target torque is done at the room temperature at 45 degrees for a period of 3 months, and the weight loss should not exceed 1% per month for the package to be considered acceptable. Then come to the leaking test. It may be advisable to test the packaged product in various orientation, upright, inverted, or its side to determine whether the packaging may leak, especially during the transport. Then safety and toxicological testing. Safety testing that includes a toxicology test. It allows them to determine if any substance of the product and the mixture present a risk when used by a customer. Several testing tests are including to highlight the product effect when in contact with the skin and the environment, eye, skin irritation, corrosion, penetration, and sanitization, eye corrosion and irritation. Compatibility testing. Compatibility test will check if there are many corrosion effects between product formulation and packaging, any alteration on product function, or any alteration on product aesthetics as a result of contact with the packaging material. The last is pH testing. The pH of the cosmetics should match with our skin pH. Normally, a skin pH is 4.5 to 7, and we check pH of the different cosmetics like cleanser. We check with the water, and we check the lip. When the cleanser mixed up with the water, then we use the litmus paper to check the pH, and water is a neutral and do not affect the pH of cleanser. This is from my side. Thank you.